This is a production of Cornell University.
Okay. Actually, one of the things I did in this presentation was try to remove most of the math. When I gave this practice talk about a week ago, there was a lot of math in there. And so I've reduced a lot of that and hopefully made it digestible. Okay, so yeah, as Jean-Luc introduced, I did my master's at New Mexico State University. I was working on auto-tetraploid alfalfa there, and particularly in transpirational efficiency. These are leaf peels, just for fun. You can see the little stomata there. And why I got, when I came to Cornell, I got interested in G by E because I had seen it, right? So oh, this thing is a little exciting when I get in there. So you can see here, this was a study I had during my master's. It's under really heavy drought conditions, and you'll notice the alfalfa that's growing right in front of it is much healthier, much greener, right? And so there are differential responses to environments. And so I'm going to briefly talk about a little bit of the work I've done with genotype and by environment interaction, mostly just as a plug for a R package I wrote. And then I'm going to move on to the part of my PhD that I'm most excited about. Okay, so typically we think of genotype by, or typically we think of a phenotype as some function of the genetics and the environment in which those plants are in. However, different genotypes respond differently to different environments. And so there's really an interaction term here, this GE, right? So there's some population mean mu, some genetic effect, some environmental effect, and then some deviation from that effect, from that additive model. Okay, and so I was interested in utilizing what's called an additive main effects multiplicative interaction, popularized by Hugh Gauss himself, and who's sitting in the audience, which is great to have him here, so he can point me out on all the things I've done wrong. So anyway, the idea is that you can think of this problem as a matrix, right? You have a table of genotypes in some environments, right? So here I've got a table of genotypes where they're the same across all environments, and a table of environment effects where they're the same across all genotypes. And then I have some deviation from that, okay? But so the problem with this G by E term is what we'd like to do is we typically want to separate out the largest sources of variability as the true, the quote unquote true G by E, and pull out the smaller sources as error. And this is particularly useful when you have unreplicated field trials. Okay, so this is done through, right, so this is a matrix, right? And what we can do is use singular value decomposition to find linear combinations of that G by E matrix that are the most associated with that G by E effect, okay? Or explain the bulk of the variance. Now, estimating how many of these we should pull out, right? So we're trying to find K of them, is another problem, especially for unreplicated data, because the G by E effect and the error are completely confounded. Okay, so I developed an R package called Bilinear that fits AMI models. It also fits other bilinear models such as the GGE and the GREG. I don't know, some of you may be familiar with these. I'm not going to talk about them in great detail. There's quite a bit of literature on this. My preferred method is the AMI because I think it's the most parsimonious. And so this package that I wrote fits various models and does some, produces plots and tests, but more importantly, has a couple of tests for how many of these dimensions we should pull out of the G by E term. In particular, we have an FR test that was developed by PIFO in '95. And this is great for replicated data, or at least data where you have a known within trial variance. But when we don't have replicated data, we can use a parametric bootstrap method that was developed by Forkman and PIFO. And so I've implemented these in this R package because at the time there were no R packages that would fit these. And I think that now there's a GGE GUI or something like that. It's a graphic user interface, but it is in R. but this fits both types of models, and it tests. And what's really important is that this can handle unreplicated data. Now, the test tends to be more conservative than the FR test is, but it's really useful, especially for our trials where we don't necessarily have replication within environment, because there's been this big shift to moving toward putting much more lines in a trial as opposed to replicating those lines many, many times within a trial. Okay, and so this is available on GitHub. I'm still uncertain. It probably needs a little bit more polishing before it'll ever go on to CRAN. But right now, from GitHub, you can install this directly in R using the DevTools package. So that's kind of fun. So these are just some examples. For those of you not familiar with a biplot, essentially what you're doing is you decompose this thing, right? And you're looking at, there's a genotype dimension and an environment dimension. So we have the first two principal components of the genotype and the first two principal components related to the environment. Okay, and so I'm not going to go into the details of how to interpret these models in particular, but basically these biplots are useful because relationships between environments or genotypes and environments are related by their angles, okay, where acute angles are associated with positive relationships, and right angles are associated with independence, and obtuse angles are negative relationships, right? So Helfer 2014 here is a very different environment from, say, Catola 16, but apparently has a very independent G/E response, whereas these sets of environments are all relatively similar to each other in the G/E response. Okay, and then we can do things like we can make these winner plots that are also out of Gaussian Zobel 97, I believe. And what we do here is we have the main effect of the genotype, and then the slope is the AMI1 fit, right? That says how they respond to a changing environment. So down here, we've ranked the environments and indicated them with these little black triangles, and then you can see that there are individuals that do particularly good in poor environments, and then their response goes down as the environment decreases. Whereas there are individuals that do particularly well in good environments, but not particularly bad environments. Okay, so this was just to kind of introduce this package. I've worked together with Lisa Kissing-Kusek, a colleague of mine in Mark Sorrell's lab. She was looking at organic wheat yield trials that were replicated from Vermont to North Dakota, looking at the response of organic grains to different growing environments. And she found that there was significant location or genotype by location effects such that it made sense to have these decentralized programs in these different regions. Now, this study was nice because it was replicated, but we ended up using the parametric bootstrap regardless because they tended to give more biological relevant numbers of interactions, so typically fewer dimensions, right? So K was low, like 1 or 2. That's in contrast to another paper I worked on with another student from Mark's lab, Lynn Veenstra. Both of these students have graduated. This one hasn't quite been submitted yet, but is very close. And we worked on G/E for fructan content, and we were able to show that there were significant G/E effects, but in general, the genetic correlation across the environments that she evaluated in New York were very similar, or was relatively high. So then we were able to recommend that breeding for fructan content could be done with relatively few environments, if only a single environment, and as well as under a genomic selection scheme. Now, this one, we did not have replicated individuals. So in this case, the parametric bootstrap was really convenient to have. Okay, now I'm going to move on to allopolyploids, really what I want to talk about. So in my second year of grad school, I was taking a course with Mark Sorrell's that I think many of you have taken. It's basically a paper review class. And in that class, we read this paper by James McKee that was published about in 1970, so almost 50 years ago. And he talks about how so he notes that whole genome duplication events are just pervasive in the plant kingdom. They're everywhere, especially in angiosperms, right? Where even if, there are these polyploid events that happened a long time ago, and then there's a bunch that have happened relatively recently too. And he talks about how there's this balance in creating new genetic variation, but then preserving particularly good sets of alleles, right? And so one thing he notes is that allopolyploids have this ability to establish homozygosity so that they can keep good sets of alleles, but they end up being heterozygous across the subgenomes. Now, that might sound a little confusing, and I'm going to clarify in a second. And that essentially this idea of allopolyploidy, bringing relatively, closely related species together into the same organism, allowed for multitudes of gene functions that should allow for broader adaptation, or adaptation in a much broader landscape, because you have these different combinations of alleles to draw from for different genetic pathways. Okay, so a little on bread wheat. So there was the A genome ancestor and the B genome ancestor. The A genome ancestor you may know today as einkorn. It's not quite the same species, but very closely related. And the B genome species is thought to be extinct, and which is unfortunate. I hope that one day we find it. Roughly 500,000 years ago, there was a spontaneous cross between these two closely related species, and they produced a tetraploid that had both an A and a B genome, right? So all of a sudden, you took a complete set of genes from one species and a complete set from another, and you brought them into the same organism. All right, and then this happened again, roughly 10,000 years ago, and maybe only once, with a related species called Aegilops tauschii, which contributed the D genome to common wheat. Okay, so now what we know of as common wheat, which is bread wheat, and primarily bread wheat, but of course also wheat for cake flour and so on and so forth, has 3 separate genomes, i.e. 3 complete sets of genes. Okay, and I've just been fascinated. What is the driving mechanism for duplicating an entire genome, right? So a single gene, we imagine, may get duplicated, and then you can maintain function with one copy and allow the other one to diverge. But what is the advantage of having complete whole sets of genes, or of genes? Okay, and so here's kind of to lay this out, right? We have 3 genomes in allohexploid wheat. It undergoes disomic inheritance, which means there's no crossing over between homeologous chromosomes. So more or less, the 3 sets maintain themselves within the nucleus. Okay, they don't switch pieces. It's self-pollinated, so it can fix itself, right? So it becomes completely inbred, and individuals will self-pollinate and produce seed that is genetically identical to the parent, right? So this is the self-preserving part, but they maintain this allelic diversity across the 3 genomes. Okay, so this is the the new generating part, right? So you grab a whole set of genes from one species and one from the other species, and now you got 2 sets of tools, right? And so I'm wondering, can we think of wheat as an immortalized hybrid? Okay, so you've brought 2. Okay, and so here's my justification for this. Okay, so in typical hybrid generation, you have some sets of alleles, X, Y, and Z, that are in an open pollinated population. They get separated from one another, either intentionally or unintentionally, into 2 heterotic groups, Group A and Group B. And then when you bring these back together to form the hybrid, you've essentially established heterozygosity at all loci, and you have these allelic interactions, which we believe may be important for what we observe as hybrid vigor, which is essentially the tendency for offspring to be better than their parents. Okay, so now think about the allopolyploid, right? We have some set of alleles in the common ancestor, and then these are a lot. These may diverge in the descendant species A and B, and then you bring them back together. But again, right? So this thing becomes inbred, and what you've done is essentially establish heterozygosity at all loci. But now you fix this heterozygosity because through selfing, right? And this disomic inheritance. So what I'm curious about is, can we see evidence of homoallelic interactions that are, analogous to allelic interactions that we see in traditional diploid hybrids? Okay, and so to investigate this, I wanted to know how this played out on agronomic traits. Now, we have a winter wheat breeding population here at Cornell. And so I kind of curated this data set. It's roughly 9,000 phenotypic records, 1,500 lines across about 10 years, 2 to 3 environments per year. And we have evaluated them for 4 traits essentially in every year. Now, this is a lot of work. It's funny how this amount of man hours can be, distilled down into 9,000, lines on a computer file somewhere. And I will definitely recognize people who have helped put this together. Okay, so this is the data I'm going to be working with to describe this investigation to you. So first thing I was interested in is subgenome, additive, and interaction effects. We typically think, okay, so coming back to this idea, we typically think of the phenotype as some, summation of all of the genetic effects, okay, on average. But there's no reason why we can't split these into their individual subgenome effects, okay? And we can estimate these using, a very standard genomic prediction model, where, and I, ignore the math. The math is not particularly important. It's just to say that here we're getting a single estimate for a breeding, for the value of every individual based on its entire genome, whereas in this model, we get a different estimate for each individual based on the 3 subgenomes. So you get 3 estimates, right? 1 for A, 1 for B, 1 for D. Okay, and this is very simple to do. We estimate the covariance between individuals using molecular markers, and so we can subset those markers based on their native genome. And then we can calculate the covariance between individuals based on each separate genome, okay, for all 3. And here's a motivating example to think of why we might be interested in this. Imagine we have this little toy population of 3 individuals, and we want to pick 2 to mate. The obvious choice might be individuals 1 and 2, which have the highest 2 values. But then if we could see, for their whole genome, but if we could see their individual effects for their subgenomes, we'll note that while they both have very good A and B genomes, they don't tend to have a very good D genome. So when we mate these guys, the best value we could possibly obtain is 20, right? Whereas contradictory, if you included the 3rd individual with the lowest breeding value, right, you have this potential to bring in, while it doesn't do very good on A and B, it does have a really good B genome, and you increase the maximum value from that cross, okay? And similarly, this is true for both of the 2 crosses with it. Okay, so, and more importantly, potentially, we can look at individuals that have the exact same breeding value and find ones that might have particularly good subgenomes that we want to incorporate for specific crosses. And so what I've done here is I've plotted the breeding values or the value that we assign to the individual based on its whole genome or the subgenome effect, A, B, and D, right? And then I've highlighted kind of the best 5% of the individuals here. And what I just want to point out briefly, the individual that has the best A genome here is not the individual with the best whole genome, nor does it have a particularly good B genome. It's very moderate for B genome. And similarly, the best B genome individual, right, has a moderate A. And so these are the blue squares. The best individual for the D genome again has relatively moderate values for the A and B. So you might say, okay, well let's take the individual's best for A and mate it to the one that's the best for B. And similarly, so in this case for grain yield, maybe most of these individuals will at least be in your pool of individuals you might select to do this, even if you wouldn't necessarily make these specific crosses. But you can see for test weight, for example, the best D individual, it doesn't have a particularly good whole genome effect. So it might be thrown out altogether and never considered for population improvement. So what this allows us to do is it allows us to select on complementary subgenomes for crossing, right? We can bring ones that have different, good effects together. Now, you could do this with any set of chromatin, right? You could estimate a breeding value for every chromosome if you wanted to. But this gets more complicated the more of these you have. Interestingly, the other thing we could do is select on subgenomes specifically, right? Like, so one of the problems is you'll notice the D genome has significantly less variance than the A and B, which means that when you make selections based on phenotypes, you are making selections on the largest sources of variance, which are coming from the A and B genome. And so a lot of the variation in the D genome is going to get fixed just through drift. And so you could select based just on the D genome, for example, for several rounds, after, say, intergressing from a wild species or something like that. But those were just individual subgenome effects, right? What I really care about is how are these things interacting with one another? And we do this through using Hadamard products of these covariance matrices. The math isn't really important. It's just that we can now get a value for every individual based on its interaction between the A and B genome, B and D, and the A and D. All right, and then we can estimate these variance components. And here, what I've shown is the different terms A, B, D, A by B, A by D, for a couple of traits. The estimates are in red with their standard deviations, and then the violin plots are distributions from sampling that data. So we subset the data, we estimate those variance components. This is actually during cross-validation. But what we can do is just see how stable is that variance estimate. We'll notice that for the most part, they align very well with their estimates. Interestingly, you'll notice there's a very different genetic architecture between these two traits. While the B genome explains most of the variance in both of them, you'll notice that here the D genome explains a lot more for plant height than it does for grain yield. And the A by D and B by D interaction effects are much larger than they are in grain yield, where they're actually very small. So grain yield doesn't have a whole lot going on for subgenome interactions, but we do see it for plant height. And then we can show that these things sum to the same variance as the full genetic effect, as well as their interactions. Okay. We can incorporate these interactions into a genomic prediction model, and we can see that when we model just the additive effects of the whole genome versus the 3 individuals and then sum them, you'll see that we get essentially the exact same prediction accuracy for all traits. And then if we incorporate epistatic interactions, one of the things we can do is incorporate all interactions, or we can just incorporate the interactions across the subgenomes. And we'll notice that, again, you see what we see increases in prediction accuracy for all the traits, except for arguably grain yield here. And that those increases are the same regardless of whether or not we're modeling all pairwise or just the pairwise across subgenomes. However, the problem is that these things are not independent, right? So in a population under pure Hardy-Weinberg, these variance components should be independent of each other. But we do have population structure. It's not like we see in something like rice, for example. But this is just a PC plot of the genetic covariance. And we're explaining maybe 18% or so of the variance, which is the first 2 principal components. So there is some population structure in here, and it's probably mostly due to the fact that we have many small families. Right, so this is done, wheat is done with pedigree selection. So this is not some big random mating, population of cattle, right? There are very specific crosses. There are few individuals from each cross that go into it, advanced trialing. There could be confounding of estimates here. And so I've had an attempt to try to remove the largest sources of variation from the marker matrix before we actually calculate how related individuals are. So basically, under Hardy-Weinberg, these variances should be independent, but typically in a breeding program, they will not be. So we can take the marker matrix and decompose it using singular value decomposition, which is obviously my favorite tool, because that's what we used for the AMI model. And we can pull out the first few principal components as population structure. I'm calling that Q. we can reform the marker matrix after we've pulled that population structure out of all of the markers at the same time. And we reform this by setting the first k singular values of d to 0 and multiplying, right? So we make this d tilde, multiply. Math isn't important again. But then we just incorporate our population structure as a fixed effect, and then we have the sum of our different subgenome effects, right? So now I can pull out this M and make a covariance from that, so on and so forth. And while having removed population structure from all three of them at the same time. Okay. When we do that, okay, so what I've done here is I've plotted in the black lines here showing the correlation of genetic relationships estimated based on the A genome and the D genome, or the A genome and the B genome, or the B genome and the D genome. And you'll notice that they tend to have a high correlation, right? And that's because individuals that are closely related are going to be closely related to each other no matter which pieces of DNA you choose to calculate that relationship. But also because of population structure. And it looks like we have this nice shelf here, right? So, you know, roughly around a K of maybe 4 or 5, it looks like we kind of hit this asymptotic relationship where the more principal components we pull out of that marker matrix, we don't get much reduction in the correlation of the relationships across the subgenes. Okay. We also take a hit in the amount of variance in the entire population, or in the entire matrix. So what I've done is, okay, so we know that K appears to be removing, right? So pulling out this population structure appears to be removing these variant components. But as I increase K, do I see big, you know, changes? And you'll notice for the A, B, and the D genomes, no matter what the size of K for these 4 traits, the additive values don't differ very much, but their interaction components do have rank changes, especially for these different for traits that are not granule. So it's really unclear whether or not we're pulling population structure out or also genetic signal, right? Because they're related to each other. What I did was I used the variance of the variance estimates to decide whether or not we should be doing this. And in general, there's not much change in this covariance. Okay, so the variance components related to the interactions tend to have a high correlation regardless of how much population structure you pull out of this thing. So in general, this was probably not, there probably isn't a whole lot of benefit to this, or if there is, it's really unclear, at least in this population. Maybe very important for certain populations that have very strict sets of population structure, like in rice, for example. But of course, that's a different. Okay, so we estimate the additive effects relatively well. It's not certain if we get good interactions. We don't lose prediction accuracy, so there's no reason not to do this from a predictive perspective, but it's unclear how you would then make selections on each individual one, right? We can do complementary subgenome selection. We select individuals that have particularly good A's versus individuals that have particularly good B's. We can select certain subgenomes. And what's great about this method is we don't need to know much about where the marker lives, only what genome it came from, which is important in crops like wheat, where for a long time we knew where markers belonged from a perspective of what chromosome they were on, but had no idea what their relative position next to one another was, because each of the chromosome arms was assembled independently. Yeah, so and this is true for a lot of polyploids. Sequence assembly is very difficult because you have because it's replicated. It's like you're trying to sequence 3 different things at the same time. So it's hard to assign who goes where. Okay. So this paper is in preparation. It's really close. I'm hoping we're going to get this one out soon. But I want to talk about explicitly modeling homologous relationships. We can consider a 2-locus model, right, where we have a locus B, say on one genome, and a locus C on another genome with additive effects alpha B, alpha C, and their interaction. Okay, and so there's different ways to think about how epistasis works. There's a classic additive by additive. I'm not going to go into this too much. This is what you get from multiplying marker scores, and anyone who's done this has probably seen this relationship. you're contrasting, the two homozygotes. It's kind of a weird thing to say because remember they're all homozygous from the heterozygotes across the genomes, right? You can look at the duplicate factor model where essentially having AB or AC gives you the effect, and having both of them does not give you any further effect, right? So if this coefficient was 2, it would be completely additive, but it's the same as having one or the other. And I'm going to introduce this idea of subfunctionalization epistasis. And this is actually a pretty, so this model is flexible. It allows both everything from a duplicate factor all the way to an additive and then beyond. And so I'm going to go into this, I'm going to give you an example of this. So you have some ancestor allele A, and it is allowed to change into the two descendant species. And when that's brought back together in the allopolyploid, you essentially get the sum of those two effects times some divergence coefficient, right? Where you can see that if D equals 1, this is completely additive. If D equals half, it's completely redundant. And if it's somewhere in between a half and 1, there's some partial redundancy. Okay, and when I say partial redundancy, I mean these genes still, to some extent, perform similar functions, such that having one or the other doesn't result in a total loss of function. But you need both of them to have complete functionality in the organism. Okay, so this is your classic epistasis plot, where you have a change from C to big C, little c to big C, change from little b to big B. I'm going to explain this using an example. So we had a biparental population where the two parents each had a single dwarfing gene. The dwarfing genes that were very famous from the Green Revolution. Essentially, if you have one of these dwarfing genes, you are what's called a semi-dwarf. That brings the height of the plant down, such that when you put a bunch of fertilizer on it, it doesn't fall over, which is called lodging. And when plants fall over and they lodge, all total grain loss. So making these guys short is good, but you don't want to make them too short. Okay. So what's interesting is this population one parent had a dwarfing gene on the B genome, the other one had a dwarfing gene on the D genome, this RHD1. Okay. Now, because we made this cross, there are double dwarfs in this population. So they have two mutant genes that are completely gibberic acid insensitive, and they are tiny. They're super short. And here we've tagged them with markers that were relatively close. You notice it's not perfect. But when we look at the mean of the population of the double dwarf, and then we can look at the effect of adding a wild type D allele, there's a linear response. Similarly, there's a linear response when we add a wild type B allele to the double dwarf. And now, based on additivity, you would expect the mean of the double wild type to be roughly here. But when we observe this, it is significantly lower than you might expect. Okay, so there's this deviation from the additive model, right? Classic epistasis, change the slope. Okay, and so this value delta is how we can estimate this directly, and we can calculate this divergence coefficient D. Okay. And then, essentially, this is this great example of 1 + 1 does not equal 2. There is some functional redundancy between these two genes. Now, this is different from there being pure redundancy, in which case you would see this, right, where this delta is exactly, where this slope is now flat. So one or the other gives you the phenotype, the wild type phenotype. And this is obviously different from seeing an increase in response. Okay, so that's one gene, but I wanted to know, do I see this pattern across the genome? Okay, and so using the International Wheat Genome Sequencing Consortium, RefSeq1, I was with roughly 1,100 coding sequences, I was able to basically establish homeoallelic gene sets, roughly 18,000 triplicates, 5,000 doubles. So triplicates means 1 on A, 1 on B, 1 on D in the expected places. And then the duplicates is 1 on A and 1 on B, but you're missing 1 on, say, D, right? So we end up with about 23,000 homeologous sets. And then I tag those sets with GBS markers, so now I can track them, okay? And I ended up with a total of about 10,000 marker sets. Now, when we look at, so what can we do? We can take these molecular markers and we can do GWAS, right? Which is our favorite thing to do here. And what I've done is I've plotted the 21 chromosomes of wheat around the circle. And then I'm indicating, these are like Manhattan plots here. So these are the negative log 10 of the p-values. And you'll see we don't have a bunch of huge hits. There's a couple. Plant height, we have the dwarfing gene on 4D. Heading date, we have PPD1 on 2D, for example. But there's not a whole lot of large effect QTL. However, you noticed from earlier, we had high prediction accuracy with these things. So there is genetic signal in there. It's just going to be, that just indicates that there's many genes we're looking at that are involved in this. Similarly, we don't find a whole lot of quote unquote significant homeologous epistatic interactions. And these are indicated by these hoops here. So, you know, a couple on 3, a couple on 4, a couple on 5, a couple on 7. Relatively few. However, we can look at the global trend of these individuals, or of these loci. Okay, so and then we can see, do they increase prediction accuracy? I'm going to get to that in a couple of slides. What I've done here is I've plotted. Oh my gosh, I'm running low on time. So what I've done is I've plotted the one homeolog, say the A genome homeolog effect, against the other homeolog, which is the, like, say, the B genome homolog effect. Okay, and then in color, I've indicated the direction of the interaction, of their pairwise interaction, and their magnitude is shown with the size of the point here. And you'll notice that we see this drastic negative trend between the additive effects and their interaction, right? So if you have a positive, if both of your alleles are positive, your interaction tends to be negative, and vice versa. Okay, and so I was like, yay, this is totally showing me exactly what I thought. This is subfunctionalization, right? But this has to do with how we coded this. So what we were doing is we were contrasting genotypes. We were trying to assign who has, you know, what is the functional copy. And this is using, you know, 0-1 marker coding. And what happens is, so to check to make sure I'm not crazy, I simulated a data set using the exact same parameters from the original, well, 4 data sets, using the exact same parameters from the original population. Only here, I intentionally did not allow them to have interactions, okay? But I didn't touch the LD in the population. And you can see we still observe this trend, and so it's like, oh man, bummer. The problem is this is a multi-collinearity problem, right? So the predictors are correlated to each other. But we can ask, are they greater than expected by chance? And so what I did was I plotted those epistatic or interaction effects from the true data and the additive only simulated data. And if there's no effect, we should not see any deviation from this diagonal line. And I multiplied them by their additive effect sign, so that if they have a negative relationship, they should have a low tail here, and if they have a positive relationship, they should have a tail here. And you'll notice for heading date, we see this push down that's indicating that these are more negative than expected by chance. We also see some for plant height down here, but we see it reflected. Not a lot for grain yield, and it's unclear for test weight. Doesn't look like a whole lot of signal. Interestingly, if I remove the LD pattern in the population, then I see this effect is to some extent emphasized. Okay, and I'll come back to that in a minute. We also, I'm going to skip over this. Okay, so we looked at, okay, so not a lot of large effect interactions, but do they increase prediction accuracy? We see that modeling all pairwise gives us this bump from the additive model, all pairwise interactions, and the homeologous epistatic interactions explain between 60 to 75% of that increase. Now, this is a very small increase, so who knows if that's real. Okay. For sanity's sake, we also sampled random pairs within and across the genome of markers, and then we looked at their prediction accuracy, and you'll see that we actually got higher prediction accuracy with these random pairs within and across, which might be like, oh man, did we miss something? why would we expect random pairs within a genome or across the subgenomes to have higher prediction accuracies than the homologous loci? If anything, I would expect them to be about the same. And the reason, I think, is we see a much higher rate of linkage to equilibrium. So here I'm plotting the linkage to equilibrium parameter d prime, so it's been standardized. And so the solid line here is the homologous set, and then the dotted lines are our two random sets. And you notice that there's an enrichment for fixation at homoallelic sites. So certain pairs of homoalleles are being fixed, okay? And so an example of this would be the dwarfing genes, just to bring it back. Most modern wheat varieties have a single dwarfing gene because they get the reduced stature that helps them resist lodging. But if you have both dwarfing genes, you're too short and you're not agronomically harvestable. So what we've done is we fixed pairs, right? We fixed pairs where you have a wild type and a mutant and a wild type. And you never have a double mutant, and it's rare to have double wild type. Okay? So this may explain why we're seeing lower prediction accuracy. Additionally, physical position is not necessarily a good indicator of LD, right? You know, it's both position and the time of those mutations that indicate LD. And I'm not going to go into this in too much detail, but there's also some difficulty in picking specific sets of markers to call homologous, right? Given that we're just using physical position to assign these, as opposed to, say, linkage to equilibrium. Now that would be really difficult to do because it's not just LD between the marker and the functional polymorphism, but also LD between the marker and the functional polymorphism on the other arm and their interaction, right? So it's not a problem for simple GWAS, right? Tagging these, and it's not a problem for all pairwise interactions because you test everything. But here we're only testing a very small subset of that. Okay, so really, so we do see these interactions tend to be less than additive for some traits. That appears pretty clear. Homoallelic pairs explain some of this increase in genetic predictability, but not more so than random pairs. And this may be due to LD or low pairing precision. So we need a better way to pair markers. And in my last 5 minutes, okay, and so this paper is essentially written. It needs to be tightened up a little bit, and we're waiting for the Wheat Genome Consortium to publish their paper so that we can publish ours. They've had the genome available for quite some time now, and they won't let anyone publish on it until they've published. So anyway, we're waiting for that. And in the last few minutes, I'm going to go into a strategy I used. So I had a problem tagging, right? So there's this question of, am I having a hard time tagging specific homoalleles with specific markers, right? They may not be in high LD. Well, what if we use more than one pair, right? So I talked about the pairwise epistasis. The problem with this is a multiple test problem. You have 11,000 markers or so. You end up with 67 million tests in a Bonferroni correction that is just really hard to see, especially for interactions which tend to only have maximized variability at allele frequencies of 0.5, right? So as those allele frequencies go to 0, you reduce the effect of the ability to see the interaction, and that variance gets partitioned to additive variance. But what if we just looked at chromosome arms? What if we took individual chromosome arms and looked at contrast to see if those arms were interacting with one another? This would drastically reduce now I'm only looking at 42 different chromosome arms for the total chromosomes of wheat, 861 total tests. So the Bonferroni correction is way less stringent. Okay, so again, essentially what I did was I took our model and I pulled out a couple of chromosome arms and modeled their interaction. And then we do a series of likelihood ratio tests to determine if the additive effects or the interaction and the interaction is significant. And then essentially we can plot the p-values, and here in light gray is the additive p-value. So is the additive effect significant? And in dark gray is the interaction effect significant? You'll notice that essentially, these would be considered significant over here. These are not. These are essentially a p-value of 1. And so you'll notice for these 4 traits, most of the chromosome arms have an additive effect, a significant additive effect, which makes sense. That's what we expect. We expect large pieces of chromatin to have effects on a phenotype. But you'll notice most of the interactions are not, which is good because we don't expect all of these things to be interacting with each other necessarily. And then, of course, when these variance parameters are estimated on a boundary, you have a problem with essentially they end up being 0. So that's where you get this big lump of p-values around 1 instead of the expected uniform distribution. Okay, and then I just want to show. So these are the results from a Bonferroni correction. What's interesting is we see some sites where it looks like we have 4D, 6A, 7D, big regulator, or a site that appears to be interacting with many loci, which is maybe suggestive of some kind of gene regulator. If we relax this cutoff a little bit, and just look at interactions that were significant at, say, 0.001, just to look at a pattern. I'm not saying that these are causing, I'm just curious. We see that this appears, we see these sites where just a bunch of interactions with a single locus or a single chromosome. Here's another one for heading date, right? It looks like it's affecting a bunch of different things. And so we see some of these hotspots, on 7D, 7B here, and 4B, excuse me, 4D. Recently, there was a paper by Jiang that came out last year, and they also saw this enrichment at 7B and 7D. So, this is essentially the same plot. Theirs is rotated. But they saw that these two chromosomes appeared to have a lot of, appear to be influencing a lot of regions. They also saw an area on 4A, which we didn't see, but we saw that 4D seemed to be important. Just looking at the homeologous chromosome arms, we don't see a whole lot of evidence for this. This is a Bonferroni correction. We only were to consider all of the ones that are significant at, say, 0.05 for their interactions. We see this. Now, you're probably wondering what the colors are. The colors are due to, I basically colored the relationship between the additive and the epistatic effect. So if they're blue, there's a positive relationship between the additive and the epistatic effect. And if they're red, there's a negative relationship. And if they're black, there's, or gray, there's no relationship. Based on just a correlation coefficient. And one of the things I'd like to point out in my last minute is that for the 4BL and 4DL here had a really strong negative relationship. And when we plotted this, you can see there's this drastic negative relationship between the additive effects and the interaction. So this might be another indicator of some functionalization. However, when we looked at this for a different trait, right? So this is for plant height. This is for test weight. We did not see the same pattern. And when we looked at our spot where we assumed we might see the interaction of the two dwarfing genes, we don't have a whole lot of them from the B genome in our population. But we don't really see much of a pattern here, although there is a significant positive correlation. Okay, so we see a lot of these interactions. Granule has a few, potentially. It looks like we see some regions that have large regulatory regions, seem to be interacting with many different loci. And we see strong relationships between some of these homeologous arm pairs, indicating that we may have some of these that there may be relationships between these homeoalleles. Additionally, I think that a lot of this stuff probably is really suggestive and probably needs a lot more fine tuning. I think a lot more research can be done. I imagine that there are some other populations that you could look at this in, as opposed to a breeding population where you fixed a lot of these interactions. You could look at a synthetic wheat population, which would maximize these interactions. And so I think that there's much more research needed into how these three genomes are talking to each other to produce a phenotype. And so at my PAG talk was in translational genomics, and I gave some of this information. And I had to figure out what the translation was, right? So my translation is allopolyploids are not diploids. For the last 100 years, we have been treating allopolyploid crops as diploids because they undergo disomic inheritance, right? So you can treat them as diploids because the chromosomes pair up and segregate, in a behaved manner. But we now have the molecular tools to start to look at these things as the immortal hybrids that they are. And with that, I'd like to acknowledge my committee members, Mark Sorrells, Jean-Luc, Yannick, who is here. Mark couldn't be here today. He is at he's at Simit, and Jason Measy. Some great folks in the Yannick Sorrells lab. I worked closely with Lisa Kissen-Kusek and Lynn Veenstra on some of the GYE stuff. I've also gotten some really good input from Tarju and Uche and Marnin and Roberto. Roberto, I will point out, was the first person that said, hey, you know, you could go in and like look at those coding sequences and then see if you could find pairs. And I was like, oh yeah, good idea. And then, of course, without the help of the folks at the Small Grains, David Venture, Amy Fox, and Jesse Chavez, and James Sanaka, who are all here, I couldn't have had this data to ask these questions with, and so I very much appreciate their work into this, as well as the relationships we developed over time. Yeah, working here. And thank you guys very much. Oh, one last one. I'm keeping you guys late. I'd really like to thank my family. They've given me a lot of support, and I couldn't have finished this without them. And I would like to thank particularly my father, who is here today, and my girlfriend Amy, who also joined us today. And thank you all. I ran a little over there, so if there's a couple of questions, sorry to keep you guys a little bit longer. Yeah.
You had one slide where your the x-axis sort of is a scalarization of the environment. It ranges sort of from -1 to +2. And
right.
It says presented. And do you think of that as an asymmetry in that measure, or is that?
Yeah, that's a good question. So I centered all of this data. So theoretically, but what I did was I can explain this. So what I didn't want to do was center the data. So this is all standardized data, right? You subtract the mean and divide by the standard deviation. Now, I could have centered within each environment, but I didn't want to do that because then you are assuming that every environment has the same variance, both error and genetic variance. Okay, so what I did instead was I centered on everything at the same time. And so then if the environment sizes, for example, like, right, there might, I don't remember which direction it was in. Was it heavier on the negative side or heavier on the positive side?
Well, I thought, I seem to remember it was heavier on the positive side. Yeah. I'm not completely sure.
So that could be from, if there were larger trials that tended to be better trials, those ones would push that whole thing, right? Because you're taking a mean. So as you summarize that thing, you're taking a single point for something that had maybe 400 genotypes in it that was a particularly good environment, and then another single point that maybe only had 200 genotypes that was particularly bad, right? So that's going to shift that mean point of the environmental. I mean, roughly they should be close to centered around 0, but they weren't because I think because I centered the data as a whole as opposed to within environment. Had I done it within environment, they should have all been smack dab at 0. Yeah.
Thank you.
Any other questions?
So I'm sort of sitting here mulling over the question about neofunctionalization within a very simple diploid, because that's both my favorite genome and mine. And I think we have neofunctionalization within duplicated genes in every even simple diploid, but we don't tend to think about the opportunity cost, because duplication is a much more conservative process than whole genome polyploidization.
Right.
And yet, if the hypothesis is true, which is that basically every angiosperm has undergone at least one round, maybe 2 or 3, of whole genome duplication, and then subsequently gain and loss of these various copies. My question is, do you see the kind of preferential loss of loci in these more, in these replete polyploids that we see in the long-term perspective on all of our ancient polyploids, which is that they actually get rid of more than half the genes that they don't want to bother to maintain, either because of your, as you've shown, they are functionally redundant, or maybe there's a cost to the neofunctionalization routines. I'm just asking that question. Is this a continuum, and do you expect to see simply the loss, and how much loss do you see when you have 3 genes?
Right. Okay. So to repeat the question, essentially, the idea is in diploid species, you have genes that get duplicated, and then those are oftentimes subsequently lost because they're unnecessary, right?
Or no, I mean whole genome duplication.
Right. Right. And so, and during a whole genome duplication, yeah, and that happens. And then, do we see that same thing happen in allopolyploids? The answer is yes. In fact, it happens really drastically, especially very shortly after the hybridization event. They usually go through this weird, this genome shock moment where they're like, oh my God, well, how am I going to deal with all of this, right? And so oftentimes big chunks, right? Because you're going to have a lot of negative epistasis too. So there's this other thing that happens where you can think of a gene from this species and a gene from that species, while they work completely fine within their own species, you bring them together and all of a sudden, like, think of like a homeodimer, for example. I think this is a good example. The homeodimers formed from the protein products within the 2 species are functional, but you might have a thing where they can still bind together and form a heterodimer across these 2 really closely related genes that may disrupt their function. So I think that when that kind of thing happens, there is a push to get, well, I don't know about a push, but there is a selective advantage to drop a lot of these really important, and we do see that. In fact, so of the 110,000 or so genes in the genome, I was only able to get triplicates for so 110,000 genome, you divide that by 3, you're roughly looking at a haploid number of 33,000 genes, right? I was only able to get 18 of them that had at least 3 copies. Or no, was it at least 2 copies? Yeah, at least 2 copies. No, I have to go look at it. I think, yeah, so there's, a third of the genome has been lost, and there was a bunch of singletons. The other thing that happens is you get a lot of tandem duplications events too. Tandem duplications are super interesting because they have a tendency to be co-inherited, whereas duplications across chromosomes, there's no linkage that holds them together, right? They can be lost very easily. Yeah. Did I answer your question? I think.
Well, I just think it's part of this picture that you're painting is that there may be immortal hybrids in the sense of these, but there's immortal hybrids in everything. Angiosperm, but the ones that have evolved longest have gotten rid of most of, they retain these duplications that seem to have an advantage, and they get rid of things that don't.
And
I'm just wondering if
and they diverge too, right?
More.
So there's this thought that essentially all neofunctionalization goes through a process of subfunctionalization, where at first they start off with the exact same function, and then you have small mutations that change them a little bit, and then eventually they develop such different pieces. And I think that this is really evident when you look at genes. Genes have these domains, right? And it's interesting that you can stick these domains together and have this thing be functional here, and that thing be, so you can it's almost like mix and match. What are those toys we play? Yeah, they're like Legos. Yeah, yeah. And so, yeah, I think it's really important, and that I would be super curious to try this in a species that is kind of in between that whole genome duplication event and already becoming a diploid, and seeing if we could find evidence of this happening in across non-homologous sites. That would be really fascinating. Yeah.
Are they lost more? Let's say, do you see more loss in the D genome, which is the new interloper, into your figure touch point? I'm just thinking about this very wide.
I think that when you bring a thing in, there's probably a lot of stuff that's good, but most of it actually has a negative effect. So there is some push to get rid of it, right? So I would assume that I should tabulate that. I should see how many of the ones I saw, where the third copy that I didn't have was from the D genome. I wouldn't be surprised if it was bulk. Yeah.
I'm going to cut you off.
Okay. Yeah.
So,
This has been a production of Cornell University, on the web at cornell.edu.